Uh-huh.
So I'm going to turn it over to Sean to introduce and take it away.
Sure. Right. So welcome to Kumaran, and it really is a pleasure to have him here today. I've actually known Kumaran for a little while now, and and I'll get to that in a second. But so Kumaran has taken a really interesting path. So he did his bachelor's and his PhD at at UCLA on the West Coast. Actually, I didn't even realize this. So he did his PhD with Olaf Schneewind, who later went to University of Chicago. And so you were there before he did his move, I guess. So with Olaf, he was working
I moved with him.
You moved with him? Oh, wow. So you can ask him about what it's like to move a lab later, I suppose. It's crazy. So while he was with Olaf, he was working on type 3 secretion in Yersinia, so a gram-negative bacterium. And so after that, he went on to do a postdoc in Boston with Rich Losick. And for those that you might know, Rich is a leader in sporulation. And so it was clear that Kumaran wanted to switch from a gram-negative to a gram-positive organism, and then work on a really cool process, essentially asymmetric cell division in bacteria, a developmental program in Bacillus Subtilis, using as a model system. And that's actually where I kind of first heard about Kumaran. So the labs in Boston that work on Bacillus Subtilis and other gram-positive organisms, they have a, I don't know, it used to be kind of a monthly meeting where they'd have supergroup and have a couple people give talks. And I remember my first interaction, well, at least the first time I heard about Kumaran's story was during one of those supergroup meetings. And he was talking about this protein that he discovered that was important for detecting membrane curvature. I hope I'm getting this right. And in doing so, helped localize proteins to different compartments of the cell. And that's actually really cool, given the fact that you're talking about a bacterium that is a single cell, and that most people think of these cells as being, you know, just bags of enzymes, but they actually have lots of compartmentalization and structure and organization to them. And so after I guess his time with Rich, he moved to the NIH, where he's been working still on membrane curvature and Bacillus Subtilis. He's dabbled a little bit in Staph aureus and cell division. They don't do the same cool asymmetric division that Bacillus does, but they're still cool. And I think today he's going to talk about some of the work he's been building, I guess, an application of what he did from his work in Bacillus Subtilis. So thanks again for joining us today, Kumaran, and we're excited to hear your story.
All right. Thanks, Sean, and thanks formally to the department for allowing me to come. So yeah, I was saying earlier that, yeah, I couldn't say no to a local institution. So hopefully you guys can see and hear me. And so, yeah, today I want to talk to you about our ongoing efforts at trying to build a totally artificial bacterial cell using entirely defined components, and then seeing if we could do something practical with this. So the bacterial cell that we want to build is not a regular old cell. It is a, let's see, how am I supposed to, there we go. Okay, it's not a regular bacterial cell. It's a specialized cell type called a bacterial spore. It's formally called an endospore, but I'm just going to call this a spore for simplicity today. And this process of spore formation is called sporulation, and we studied this, as Sean mentioned, in this model organism called Bacillus Subtilis. So this is what Bacillus Subtilis looks like if you look at it under a regular light microscope. This is in the presence of plenty of nutrients. It's a rod-shaped bacterium. It does what other bacteria do. It divides right down the middle to create 2 daughter cells, and then they go on to create 4 daughter cells, and this keeps going on and on. Nothing too special here. But what's amazing is that if you introduce these cells to a nutrient-deprived environment, when they eventually run out of food, they'll completely freak out. They'll spend the next 6 or 8 hours transforming themselves to metamorphose into a completely different cell type called an endospore. And that's what you're seeing on the right-hand side. So, you know, obviously these two cell types don't look at all similar, but I assure you it's the same organism. So we've been studying, this process of how the cell on the left-hand side turns into the cell on the right-hand side. So here's a schematic that's based on about 40 years of literature of how this proceeds in Bacillus subtilis. So as I showed you, Bacillus is a rod-shaped bacterium. The first event that happens when it reaches a nutrient-deprived state is that it senses this, and instead of dividing down the middle like it normally does to create two genetically identical and phenotypically similar cells, it'll asymmetrically divide as shown here. And this immediately creates two different cell types. There's a smaller cell called the forespore, and then a larger cell called the mother cell. And what I should point out here is that they both contain the same genome. So they're genetically completely identical, but as you're going to see in a moment, they're going to have different cell fates. So the next step in sporulation is that bigger cell, the mother cell, begins to swallow the forespore in a process that we call engulfment. Now, eventually what's going to happen is that leading edge of the forespore is going to pinch off, and that will pinch off now the forespore so that it exists as a double membrane bound organelle inside the mother cell cytosol. So this is a little unusual, mainly because bacteria usually don't have intracellular organelles like eukaryotic cells do. So this is a rare example of a bacterial organelle because it's a membrane bound over here and it has its own genetic material on the inside. So now from this point forward, the process keeps going on as the big cell feeds the small cell. Eventually, the mother cell will lyse. That's a programmed event. It's supposed to happen, and that's the fate of this mother cell over here. It eventually lyses. And now this releases the now mature spore into the environment. And so here it'll stay. It's a dormant cell type, and it's one of the most hardy organisms on the planet. It's going to be resistant to all sorts of environmental insults, things like high temperature, desiccation, organic solvents, you name it, it can probably resist it without dying. Until growth conditions are favorable again, at which point the cell will spring back to life and then start growing as if nothing happened. So just as an anecdote, I can tell you that there are different time points that people have estimated that these things can survive for. Some of the ridiculous estimates is that they can last for millions of years. Some of that is being questioned by different people now, but definitely they can last decades and perhaps hundreds of years. And some of the more outrageous theories is that life on the planet could have actually originated by spores from other planets that survive space radiation, for example, which these spores do, and you can see life on the planet this way. That, to my knowledge, is so far unproven, but yeah. The reason I bring up all these sort of resistance phenotypes is because this is partially mediated by a very thick shell that's built on top of these spores that's called the spore coat. And so our lab has spent the last several years studying how this huge complex is built as a model system for understanding how localized protein structures are built inside a cell. So this is what that structure looks like. So this is an electron micrograph of a negatively stained thin section of a spore. And what you're supposed to see here is that this area highlighted in green is the spore coat. There are about 80 different proteins that make up this structure. And what I'm showing you here is that hopefully you can get an appreciation that these 80 proteins are not just sort of vomited onto the surface of the spore, but there's actually some elegant architecture here. So for example, there's this inner lamellar layer over here. There's this, yeah?
Can I interrupt for a moment? So
Sure.
A couple of things. So one, people can't see the electron micrograph that you're trying to show right now.
Oh, that sucks. Okay.
It can pop up.
This might be a problem.
There it is. Perfect.
Okay,
Now
good.
I can see it. Then there was just a question while we're paused here. So Leslie was asking about how high temperatures do these spores survive?
Yeah, so Bacillus subtilis, the lab bacterium, can routinely survive 80 degrees Celsius. You can probably push it even higher than that. There are some extreme examples out there of ridiculous hyperthermophilic heat-resistant spores.
Perfect.
Mm-hmm.
Thank you.
Yep. Yeah, and actually, you know, now spore remediation is a big problem because you know, you got to get rid of it, especially for, you know, places like NASA. When you send things up into space, you got to get rid of these spores so that you don't contaminate foreign environments. So there have been, for example, specific species that are really hard to kill that have been isolated from the space shuttles, for example. So yeah, so this is the coat. Yeah, so there's this electron dense outer layer, and then there's this fuzzy stuff on top. So this is just to let you know that, you know, there's an ordered structure in this. So this is the entire punchline of what I'm going to present over the next hour or so. Okay, so over the last few years, we've come up with this model that is very painfully summarized on a single slide that tells you what we think, how we think this complex structure initiates its assembly. And this entire thing happens with just 2 proteins. And these are the only 2 proteins that you have to keep track of in today's talk. Okay, so, and I think they're mostly color-coded going forward. So the whole process starts off with a protein called SpoVM. This is a Roman numeral. I'm just going to apologize for the clumsy nomenclature for sporulation proteins, but it's not my fault. So SpoVM localizes to this surface. It's made in the mother cell cytosol, and it localizes specifically to the surface of the developing forespore. The second step is that it recruits another protein shown in green called SpoA. That's also produced in the mother cell cytosol. It gets recruited to the surface of the forespore. And then finally, that second protein, SpoA, will polymerize on the surface, creating a tough basement layer on top of which the other 78 or so other proteins will come and assemble. Okay, so today's talk, I'm going to talk about how that first protein, SpoVM, localizes properly. Second, we're going to talk about how the basement layer of the coat is actually built. Third, we're going to see if we can assemble the basement layer of this coat in vitro, so artificially, to build these synthetic spore-like particles. And at the very end, we're going to see if we can use these synthetic spore-like particles to deliver drugs to specific tissues in the body. Okay, so we'll start off with this first topic of how does SpoVM localize properly. So what I'm showing you here is a fluorescence micrograph of sporulating Bacillus subtilis. So to visualize the cells, we've used a fluorescent membrane dye that's shown here in red, and I'm highlighting it right now, and that shows you the outline of the cells. And these cells have been engineered to express that first protein, SpoVM. It's fused to a protein called green fluorescent protein. So this allows you to visualize cells that they're attached to. And what we can see here is that this protein is able to localize very specifically to that surface of the developing forespore. I want to introduce another variant of this protein, and these are the 2 tools that we started off with. So on the right-hand side, I'm showing you the same protein, but instead we've changed one single amino acid in that protein. It's an amino acid called proline at position 9 that's been changed to another amino acid called alanine. If you change that one amino acid in this protein, it does this remarkable thing that's on the right-hand side, which is that it still goes to the forespore surface, as you can see here, but unlike the regular or wild-type protein, as we call it, this variant mislocalizes to all the available membranes in the mother cell cytosol. So this protein on the right has lost its way just because we changed one amino acid. So a few years ago, we solved the protein structure of SpoVM. So what I should point out to you is that SpoVM is very unusual. It's a tiny, tiny little protein. It's only 26 amino acids long, and it's an amphipathic protein that embeds into the membrane. So what I'm showing you here is the structure of this protein that was solved in collaboration with Fang Tian's lab at Penn State University using a technique called nuclear magnetic resonance. But the point here is that this is the structure of this protein as it resides inside a membrane. And the orientation of this protein in the membrane is shown on the right-hand side. So this is an all-atom molecular dynamics simulation of this protein sitting in a membrane bilayer. The take-home message here is that, number one, the protein is located, unlike other membrane proteins that span the entire length of the membrane, this one actually sits in one leaflet of that phospholipid bilayer. So just to orient you here, this is one leaflet of the phospholipid bilayer. The head groups of the membrane are labeled in purple up here and down here. And then what are called the acyl chains, the hydrophobic part of this, the greasy part, is shown over here. And you can see this delineation by color of the two leaflets. And so this protein just sinks itself into one leaflet and is localized so that it's parallel to the plane of the membrane bilayer. So this protein, you can see, even though it's very small and simple, still has a structure. So there's this floppy N terminus, and then there's this alpha helical C terminus. And both of those domains are delineated by that proline residue at position 9 that I mentioned to you earlier. And that sits right at that intersection between these two domains. And we also solved the NMR structure of the variant that I showed you earlier that doesn't localize properly in the cell. So this is that proline that's been changed to an alanine. And what you can see immediately is that the structure looks different. So that floppy N-terminal region now is flattened out, and the whole protein just looks like a normal, boring sort of alpha helix. So immediately then, with the structural data, we had the structural basis for why one protein acts differently than the other protein inside the cell. So these are the two tools that we're using, and you're going to see this protein used as a negative control in several of the following experiments. So, we have this idea for, how does this protein, the regular protein, localize to this surface specifically and not localize to any of the other membranes that are available inside the cell. And so a few years ago, we had this very nutty hypothesis that perhaps what SpoVM is recognizing on this surface is not something like a pre-localized factor, but maybe it actually recognizes the shape of this membrane. So this is the only surface in the cell that has a convex or positively curved membrane. So this is like the surface of a basketball. The other membranes that are inside the cell are all concave or negatively curved. So that's going to be like the inside of a salad bowl. So our hypothesis was that this protein somehow is able to recognize the surface of a basketball versus the inside of a salad bowl. So how do we go ahead? I mean, it's a crazy hypothesis, but it was testable. So we wanted to go ahead and start testing. But before I start and give you the test, I just want to give you an idea of scale here. So the problem is that this protein SpoVM is tiny. It's about 40 angstroms if you consider it as a perfect 26 amino acid alpha helix. And this protein sits on top of a sphere that's the surface of the forespore that's about 1 micron in diameter. So you can't see much here, so I'm going to zoom in on this. And what you can see here is that, so if you do some quick calculations, the difference in membrane inflection from one side of this protein to the other is about less than 0.2 angstroms. So this is basically the difference in whiteness that you see here in the scale drawing between this side and this side. To put this in perspective, a carbon-hydrogen bond is a whopping 1 angstrom. So basically, this is like you standing on the surface of the Earth trying to tell if this is curved or not. So this is the challenge that we're facing here, but we wanted to go ahead and test this hypothesis. So I'm going to show you one simple experiment initially where we tested this, where we used a previously characterized mutant of Bacillus Subtilis that's blocked at this stage of sporulation. So basically the idea here is that there's no convex membrane surface anywhere three-dimensionally, if you imagine this, in this mother cell cytosol. So our prediction is that in this mutant cell, this regular protein should mislocalize to all the available membranes. And so here's what we saw. So this is now in a regular cell. I showed you that SpoVM fused to green fluorescent protein localizes to the surface of the forespore. But if we look at this mutant down here, this is the membrane stain. You can see that the septum that's initially created is perfectly flat, and you don't get that curved membrane. And if you look up here, this protein now, you can see, goes to all the available surfaces that are available in this mutant. Okay, so this is consistent with the idea that the protein will preferentially go to membranes that are convex and not, either flat or concave. So that was great, but we needed a robust assay biochemically to understand how this protein could preferentially absorb to differently curved membranes. So to do this, I want to introduce you to Jean-Philippe Casting. He's a former postdoc that used to be in the lab. He has now returned to his native France, and he works at a biotech company as a project director. So what Jean-Philippe did was that he did this biophysical assay where he starts off with silica beads that you can buy commercially that are available at different diameters. So he chose either 2 microns in diameter or 8 microns in diameter. So I just want to remind you that the Bacillus forespore is about 1 micron in diameter. So this is close to the curvature that you should be seeing on the surface of the spore. And this is less curved. And then he took these beads and he incubated them, or he laid down on the surface a membrane. So basically a phospholipid bilayer. He then purified SpoVM fused to GFP separately. He mixed them both together, and he wanted to see how this protein in just a regular solution would absorb to these differently curved membranes, and then he measured this using fluorescence microscopy. Okay, so I'm going to show you the data backwards because what I'm going to show you first is that variant of SpoVM that I showed you earlier mislocalizes inside the cell. So if you change that proline to an alanine, this cannot distinguish curvature inside the cell. And what he noticed was that at low concentrations of the protein, this mutant protein goes to the big beads that are inside the mix, and it also goes to the small beads that are inside the mix. It doesn't care how curved the surface is. And if you increase the concentration, it just increases the amount that it absorbs onto each differently curved membrane. Okay, so now comes the killer experiment. He did the same thing with regular SpoVM fused to GFP. Now, when he incubated that with the differently curved membranes, he saw something completely different. So what the arrows are pointing to here is the absorption of the protein seen by microscopy to the smaller beads that are in the mix. And what you'll notice is that right over there on the bottom, you can't see anything, but there's a bead that's hiding there that's a large bead. What you'll notice is that there's hardly any of this protein that's absorbed onto that bead. Okay, so what this told us was that at certain concentrations, this protein seems to choose more curved membranes than less curved membranes. And then you can increase the concentration until finally you run out of room on the small beads, and then eventually it'll go to both beads when it reaches saturation, as we call it. Okay, so we need to go ahead and quantify all of this data. And it's hard to do in a rigorous way with microscopy. So we came up with another method of trying to quantify these data. And I want to introduce you to Eddie Kim, who's also a former postdoc in the lab. He's now a data scientist at Vivo Incorporated. It's a music video company in the Bay Area where he's a data scientist, where he's busy looking at all of your personal data. I should point out that he actually got 2 assistant professor positions offered to him. He turned both of them down and decided to do this thing off in the Bay Area, and I supported him in that decision. Okay, so what Eddie did was to use this method called flow cytometry. So basically what you're doing here is you take that mix of proteins absorbed onto these differently sized beads, and then you run it through dripwise through a fancy machine called a flow cytometer. And what this machine can do is separate out big beads shown here from small beads by using a technique called forward and side scatter. It can then, as it's separating out the size of these beads, also look at the fluorescence intensity of each one of those bead sizes. And so in these experiments, Eddie, for technical reasons, didn't use the GFP fusion. Instead, he's using a small molecule that's fluorescent called Cy3. And so whereas the microscope-based assay lets you look at maybe dozens of beads, this will let you look at about 30,000 beads in a reaction volume. And when he looks at that, you can actually go ahead and quantify the amount of fluorescence or absorption that you're seeing onto the big beads shown in red versus, the small beads shown in red versus the big beads shown in blue. And what he noticed was that, as you increase the concentration of the fluorescently labeled protein, the amount that binds to the small beads rises very quickly until it runs out of room. And if you look at how it absorbs onto the big beads or the less curved surface, it rises less slowly. And this is classic, a classic example suggesting that something has a greater of what we call affinity or intrinsic sort of stickiness to one surface versus the other. And so you can quantify this using this value called the K-half value. So basically you're looking at the concentration at which you receive, which you get half maximal absorbance. So if we look at this value for the 2 micron beads, the more curved surface, it's a smaller volume, suggesting that it has a higher affinity than it does for the larger value for the 8 micron beads. Is that clear? Okay.
I like it. It's I've got one of my biochemistry students in here, so he'll appreciate the K-half values that are there.
Outstanding, because it's going to get even more complicated.
One question before you move on.
Yes.
So Tom Coat is here, and he's asking whether or not SpoVM aggregates. He's talking about how the spheres appear to have some puncta, so he was just wondering about if they aggregate or not.
That's gorgeous. Okay, so yes, so I got to say this. So purifying this protein is not trivial. As you imagine, it's very hydrophobic. So basically, we got a way to be able to purify this amphipathic protein so that we can get it soluble to some level in the absence of detergent, which is required to keep the membrane sort of intact, because you can't put membranes with the detergent, right? So that said, when you add the protein to the membranes, we did notice that at low concentration, as you did too, that you actually get 2 ways of sort of aggregation, if you will. So number one is on some beads, you'll see sort of localized increase of fluorescence at certain patches. So that's number one. And number two is, as you keep going on in concentration, if you actually look at some of these bead populations, you'll see some beads have the protein, and then some beads will be completely devoid of any protein at all. So it almost suggests that there is a seeding activity where some of the protein sticks and then recruits positively subsequent binding events onto the small beads. And this is almost binary because some of them haven't initiated that yet and some beads have. So biochemically, again, if you look at the binding curve, this is usually, so this is a phenomenon called cooperativity, where the binding of one event positively affects the next binding event. Biochemically, if you look at a binding curve that looks like the one I'm showing you right now, that is reflected in the shape of the curve, because you get a sigmoidal shape. It looks like an S. Now, if you look at this, you'll actually notice that the binding of the 2 micron beads in red is actually more S-shaped than the 8 micron beads. There's a coefficient that some of you have probably heard. It's called the Hill coefficient that describes that cooperativity. And this 2 micron beads does have a higher cooperativity of binding than the 8 micron beads does. So yes, there is a cooperativity component. I'm not going to get into that too much today because of time, but I can tell you that that cooperativity component we know is mediated by that floppy N-terminus of that SpoVM protein.
That's so cool. Thank you for clarifying.
Sure.
There was one other thing before you move on, and Anne and I were thinking about the same thing, I guess. So clearly it will mislocalize if the levels get too high, and so in the cell, this must mean that they must be controlling the expression of the SpoVM gene exquisitely to do this right.
Yeah, so I can tell you this, right? So, okay, so the control of the gene happens where, okay, it's only expressed in that one compartment. So there's that. There is the copy number of this gene, the protein is really high because you need to cover the entire 1 micron surface of that forespore with this protein. So I think it is an accidental thing where, you can just go ahead and make a ton of protein and you still will have room on that 1 micron surface to shove all of it onto there before the other membranes come into play.
Got it. Cool.
Okay.
And then Mark also, well, there's actually a couple more questions coming in.
Sure.
Mark, did you want to unmute and ask your question directly? You had a comment. I don't know if it was answered or addressed already with Tom's question.
Yeah, it was actually a response to Tom, but it does make me wonder, are there mutants that I guess your mutant in the proline doesn't doesn't polymerize, is that right?
Okay, so polymerization is a loaded term, so I'm not going to use that. I will say that the proline, and I'm not going to show you the data for this, but we've published it. So the proline mutant exhibits far less cooperativity than the wild type protein does. So there's less evidence of that. So I'll get to this a little bit later where I hand wave about this. So, you know, cooperativity usually means that you have protein-protein interaction. And so, you know, to recruit the second one, you need some sort of physical touching. There's no evidence that we have that SpoVM directly contacts other molecules of SpoVM. That said, there could be an apparent cooperativity that's exhibited on the surface of a membrane once you have adsorption. And that basically happens through the membrane. So, I mean, I can get into this really quickly where, you know, if you have a protein that binds, there's what they call an energy cost associated with that, where you deform the membrane locally. And then if you have another binding event somewhere else, you have another deformation there, and that comes with an energy cost. And it turns out that you can minimize the total energy cost of the system if they spontaneously cluster towards one another. So that's usually a phenomenon called entropic clustering, which we think might be going on here. So you guys see this every morning probably. So if you pour Cheerios into a bowl of milk, and if you have a limiting amount of Cheerios in the bowl, you'll notice that the Cheerios will spontaneously cluster together. You'll never see a situation where the Cheerios are all over the, like if you have 5 or 6 of them in your bowl, they'll always cluster together. This happens with soap bubbles in your sink too. So we think that that might be a phenomenon here, and that's where the cooperativity could come from.
So where does the curvature, how does the curvature of the membrane affect that?
Outstanding question. So we don't know yet. So I'm going to give you my best guess is that we think that the packing of the acyl chains of the lipids are different in the differently curved membranes. And we are actively trying to show that now.
Which gets at a question that Jeff Wong was asking about whether it has preference for lipid type or is curvature sufficient. So
yeah, so as far as we can tell, the lipid head group type does not matter. So we've done these experiments where you can, you know, so let's see. Actually, these experiments were done with synthetic lipids. So it's it's PE, PG, and cardiolipin. So mimicking E. coli lipids, but with synthetic lipids. We've done this with DOPC, for example, phosphatidylcholine, which pretty much isn't found in bacteria. And you can still see this phenomenon. So my short answer there would be there may be a lipid contribution to how SpoVM localizes, but it certainly isn't necessary. The curvature seems to be sufficient.
Got it. Cool. Looks like that's all the questions for now. And just to give you a sense of your time, just because in case you can't see your clock, it's 1:06.
Okay, so what am I going to?
1:30-ish.
Okay, so I'll go through this quickly. So this affinity portion of this can be described as a ratio, okay, as an off rate and an on rate. Okay, so the idea was that, remember I told you, if affinity was, if that number was low, it means you have a high affinity. So basically, if you have a low off rate, if you sort of stick to something and then never come off, your affinity number is going to be low, which means you have a high affinity. So we wondered, for example, then, does a slower off rate underlie this increased affinity of SpoVM for convex membranes? Okay, so usually this happens at like a millisecond level. So it's very difficult to measure on rates and off rates. But there is another aspect of this that we can exploit. So this off rate can be described as an inverse of what's called residence time. So basically, if you spend a lot of time as a molecule on a surface, that means that your off rate is going to be very low. Okay, so it's just the inverse, which means that your affinity number will be low, which means your affinity is very high. I'm telling you all this because this residence time is something that we can measure using single molecule fluorescence microscopy. So our idea was to measure this residence time on differently curved membranes using a fluorescently tagged SpoVM to site 3. So these experiments were done by Emily Peluso. She's a former post-bac student in the lab. She spent 2 years in the lab. She's now a graduate student at UCLA. So Emily came into the lab with no experience with biophysical techniques, but she has a big brain. And so she said what she wants to do is to affix these membrane-coated beads of different diameters on a cover slip. She's gonna then come back with SpoVM that's fluorescently labeled. And then she's gonna image limiting amounts of this. So we put tiny amounts of this protein into this mix, and then she's going to image these individual molecules of SpoVM Cy3 using a technique called high-low microscopy that shoots the excitation light at an angle like this to reduce background. So we did this in collaboration with Hari Shroff's lab at the NIH, and they insist on me showing you their intimidating setup, their microscopy setup that they use to do this. Okay, so here's the data. So hopefully you can see this video that's going on. So on the left-hand side, this is just a bright field image of the, what looks like, 4 or 5 beads on this cover slip. And on the right-hand slide, what you're supposed to see are white spots that appear and then they disappear. Okay, so I'm not going to show you all the controls for this, but basically with this, when you see a dot appear, this represents a single molecule of SpoVM, fluorescently labeled, sticking onto the surface of a bead. And then when you see it disappear, it is not a phenomenon called photobleaching where it just gets bleached away, but it's actually the protein jumping off of that membrane afterwards. So you can go ahead and quantify all the data of all these events that occur on the surface of each one of these beads, and you can quantify it. And so we went ahead and did that, the quantification of the protein doing this on the big beads and the small beads. And what we see for the wild type protein, the regular SpoVM, what we see is that it displays a longer residence time on the more curved surfaces compared to the residence time that it displays on the less curved surfaces. And so this gets at one of the questions that was asked earlier: what happens to that mutant that mislocalizes or promiscuously localizes to all the curved membranes? And what we see is that this difference between the two curves membranes is drastically diminished. So taking all this together, we've come up with this, what we call a dash and recruit model for curvature recognition by SpoVM. So in this model, the protein, which is normally disordered when it's in solution, binds onto the differently curved membranes. And this we think is primarily dictated by an intrinsic affinity difference dictated by a longer residence time for the protein on more curved surfaces. Like other amphipathic alpha helices, SpoVM only assumes its particular shape when it goes into the membrane, after which it inserts. What we're arguing is that when this insertion event happens on the wrong curved membrane, the protein bounces off and resulting in a longer residence time of the protein on membranes of proper curvature. This is where that second component, where I didn't touch upon too much, called the cooperativity, comes in, where this protein then recruits other proteins to the similar surface, and then this basically takes off to the races. And we think that this cooperativity is mediated by flexible N-terminus. And so this is just to show you that, so this may not be this, this recognition of micron scale membrane curvature might not be limited to this obscure bacterium, but there's now some evidence in eukaryotic cells that the septin cytoskeleton may also employ a similar strategy in recognizing slightly curved membranes on this level. Okay, so the second part of the talk I want to talk about was the how the basement layer of the coat is built. So to do this, I wanted to briefly remind you of cytoskeletal proteins in biology. So for the purposes of this talk, there's two kinds. There's the dynamic cytoskeletal proteins, things like actin and tubulin. And what they have in common is that they'll go ahead and bind to a nucleoside triphosphate, either HTP or GTP. And what they have in common is that when they bind to the NTP, they'll polymerize. And then when they hydrolyze the bound NTP, that hydrolysis event is linked to the dissociation of that dynamic polymer. And that makes sense because these are supposed to build and then get disassembled. On the other hand, you have these static cytoskeletal proteins called intermediate filaments. These are things like keratin on your fingernails. What they have in common is that they spontaneously assemble without the requirement for a nucleotide. And pretty much once they assemble, they'll stay assembled and they won't disassemble again. The reason I point out all this is because that second protein, SpoVM, that's recruited by the first protein, a few years ago, we noticed that it binds and it hydrolyzes this nucleotide called ATP. And what was interesting is that if you purify the protein and incubate it with ATP, but not with a non-hydrolyzable form of ATP. And if you look at that under an electron microscope, this protein seems to form these filaments. And this is interesting because this non-hydrolyzable form of ATP will not form this filament. But if you incubate, say, actin with ATP gamma S, it'll form that filament just fine. So what this told us was that maybe polymerization, unlike, say, actin or tubulin, requires nucleotide hydrolysis and not simply binding of the nucleotide. So this is what the protein actually looks like. So this is a predicted topology diagram of the protein. So there's this N-terminal protein that binds to the nucleotide. We solved this in collaboration with the Arvind lab at the NIH, where there's a second domain that's been delineated that we think is largely structural. We creatively call this the middle domain. And then there's the C-terminal domain that we've shown experimentally. This very tip of that C-terminus interacts with that first protein, the curvature sensing protein, at the surface of the forespore. So I'm going to zoom in on this nucleotide binding domain that's right over here. So this is where the nucleotide sits. The base is over here, and the 3 phosphates sit along here. And when we did this sort of analysis, we can immediately locate a so-called Walker B motif. So the aficionados in the audience will recognize that this is a motif that coordinates a magnesium ion that's required for nucleotide hydrolysis. So I'm not going to show you the data for this because it's already published, but we showed that if you disrupt this Walker B motif, this mutant protein will bind ATP just fine, but it won't hydrolyze ATP, which is exactly the mutant that we were looking for, and that's the mutant that we're going to be using in the following experiments. So this is Jean-Philippe Casting again. So he wanted to measure the kinetics of SpoVM polymerization. So to do this, he took the purified protein, labeled it with a fluorescent dye, incubated it with ATP, and then measured the accumulation of the insoluble polymers over time using turf microscopy. So that data is shown here. So this was done in collaboration with the Sellers lab at NIH. So at T0 over here, we're looking at the purified protein, and you can't see very much on the microscope cover slip. But if we wait long enough, we can see that the regular wild type protein assembles over time. If we now do this experiment with the variant of the protein that binds to ATP but doesn't hydrolyze it, we can keep looking there for as long as we want, but nothing appreciably polymerizes over time, suggesting that this protein requires ATP hydrolysis and not simply binding to polymerize. Okay, so the next thing we wanted to check was, okay, so is this ATP, the polymerization of SpoVM reversible? So remember, this nucleotide binding activity is usually relegated to dynamic cytoskeletal proteins that polymerize and then depolymerize over time. So to test this reversibility, we ripped off a classical experiment that was first performed for tubulin to show the dynamic instability of tubulin. So what we did was we took the purified protein, polymerized it in the presence of ATP overnight. The next day we came back and then diluted the reaction threefold. So this was below the threshold concentration of polymerization that we experimentally determined. So basically, if you do this with tubulin, the tubulin polymers will fall apart and they won't be able to reform because they're now below the threshold concentration for polymerization. And so then we went ahead and measured the polymerized SpoVM after time using differential centrifugation. So this is what we saw. So at T0 here, this is right after dilution of the entire reaction. And what you can see is that we recover most of the material that we started off with. What's amazing is that if you now wait many hours, even 24 hours after we diluted, we're still able to recover most of the polymerized material that we started off with. And what this suggested to us is that once SpoVM polymerizes, it's largely irreversible. So it's not a dynamic polymer. Once it's polymerized, it stays polymerized. And this makes sense because this is forming the basement layer of that structure on the surface of the forespore, which is supposed to be very sort of static. Okay, so taking all of this data together and also new data that I haven't had a chance to show you yet, here's a model of how we think this protein polymerizes. So this protein starts off as a dimer. It binds to ATP. We've shown that the binding event alone causes a conformational change in the protein that we can measure by a technique called limited protein or trypsin proteolysis. After the binding event happens, the bound ATP is hydrolyzed. And what we've recently shown is that that hydrolyzed ADP is actually held on briefly as the protein multimerizes. What I want to point out, though, is that this is not a polymer. This multimerization intermediate is actually stable, but when you, under low concentrations of the protein, but if you increase the concentration of the protein artificially, what we found is that the ADP pops off and you get a nucleotide-free polymer, and that's what we're actually detecting by EM. So in vivo, this is what we think is happening. This protein is made in the mother cell cytosol. It quickly undergoes these conformational changes because there's plenty of ATP in the mother cell cytosol. But once it gets to the surface of that forespore, recruited by SpoVM, we think that that causes a local concentration increase in SpoVA, which allows it to polymerize at the correct place and the correct time. So not to beat a dead horse here, but we think that now SpoVA, because of this unusual nucleotide requirement, represents a different class or category of cytoskeletal proteins in biology. So now we've got these two components. You got the first protein that localizes by curvature. The second protein comes in and hydrolyzes in a very unusual way. We wanted to know if we can go ahead and assemble the basement layer of the coat in vitro to build these synthetic spore-like particles. So this is Elin Wu. She's a former postdoc in the lab who was trained as a chemist, who's now working for Thermo Fisher in the Bay Area as a scientist. So what she decided to do was to combine all of the reactions that I showed you earlier together in one test tube. So she starts off with those silica beads that I introduced to you earlier. She coated it, her strategy was to coat it with a phospholipid bilayer. She comes back with a synthesized SpoVM peptide. So remember, it's only 26 amino acids long, so we can just synthesize this, absorb it onto the surface. She then purified SpoVA, and then in these experiments, she's incubating it with the ATP. In these experiments also, the SpoVA is labeled with a fluorophore, so she can visualize it. And the idea is that it should polymerize on the surface of these beads. And that's exactly what she saw. Okay, so these are the experiments where that SpoVA is actually labeled. And in the presence of ATP, we can see that the protein specifically polymerizes onto the surface. And the idea is that, at least qualitatively, this is supposed to mimic the localization pattern of the protein that we see inside the cell, where it seems to localize to the surface of the developing spore when it's fused to green fluorescent protein. So, we have this biophysical problem here where, SpoVM, I told you, is a peripheral membrane protein that comes on and off of the membrane. And then the second protein comes and polymerizes, but how does this then stably recruit SpoVA at the right place without, getting this promiscuous localization of both proteins everywhere? So to do this, we came up with this model that we call the two-dimensional Ratchet Model that we recently published. So in this model, the idea that we used to sort of wrap our heads around this was that you have a finite number of binding sites on the forespore surface that we can imagine is like a square lattice of binding sites that we can envision. So the idea is that SpoVM could localize to, the first protein can localize to any one of these binding sites. And I showed you earlier that this reaction is actually reversible. So you can bind onto those, each binding site, and each one of these ones can pop off at a certain frequency. We envisioned then that the localized SpoVM could recruit the second protein, SpoVA. Okay, so this is a protein-protein interaction. So as you can imagine, this is also reversible. Okay, so the recruited protein can also come off. But I told you that SpoVA, once it's absorbed onto the surface of the forespore, will then polymerize because that's where the concentration is the greatest. Now here's the kicker, and this is why we call it a ratchet model. We proposed that this polymerization process is a one-way street. And once it polymerizes, none of these proteins can now pop off of this membrane. Okay, so if you have a model like this, you can actually propose that it's a quantitative model because you can assign what are called rate constants to each one of these steps that you can individually measure. Now, earlier during the talk, I actually showed you what the on rate was of SpoVM, and I told you that we can also measure an off rate for this. Similarly, we went ahead and used a single molecule strategy where the fluorophore was on SpoVM, and we were able to measure a on rate and an off rate of SpoVM, and I'm not going to show you the data for that because it's already published. But what I will show you is that this last rate constant, our model here is that once this the recruited protein was polymerized, then nothing comes off of that membrane. So we wanted to test if this is actually an irreversible step. So the first thing we did was an in vivo experiment where we looked at wild type sporulating cells, and this is the localization of that first protein, the curvature sensing protein. It goes to the forespore. If it's true that the second protein prevents the first protein from coming off the membrane, if we got rid of that second protein, we would expect that now the first protein should mislocalize. And that's exactly what we see. So if we delete that second, the gene encoding for that second protein, we can see that that first protein, mostly localizes where it's supposed to go, but it also comes off and it's seen in the static image at the wrong membrane. And what we're interpreting is that this is the protein sampling different membranes. So we wanted to see if we can actually see this in vitro in our system. So this is that same bead-based assay that I showed you earlier, where the first protein, the curvature sensing protein, is labeled with a fluorophore. And if you add the second protein and ATP, at an early time point, you can see that the first protein bounces on and off of that of these beads. Okay, and that's evidenced by that blinking. If you, however, wait long enough, this is what you see. That bouncing around no longer happens, and that first protein is now locked into place. The reason that you're seeing this fade away is a phenomenon called photobleaching, because you keep blasting the fluorophore with UV radiation. Okay, so so that's now going away. Okay, so we wanted to make sure that this was not just an in vitro artifact or something, so we did the same single molecule localization of the first protein in vivo inside sporulating cells. So what you can see here is that, at an early time point during sporulation, the first protein fused to a halo tag is sampling different membranes. But if you wait long enough, what you see is that it's been locked on to the surface of the forespore. Those proteins are not moving very much, and every once in a while you're getting a sampling of the membrane by single molecules of the first protein. So this suggests that that second protein is actually locking the first place onto the surface, and using these sort of experiments, we can actually measure a rate constant to describe this. So now that you have all these rate constants, you can go ahead and run a deterministic simulation, and that's what's shown here, using all of these in vitro derived rate constants and in vivo derived protein concentrations to reveal what's actually a very dynamic code assembly process. So in these experiments, we're looking at multiple populations that I'm going to be telling you one by one. And if you look at the first fraction of the first millisecond of the simulation, what we notice is that the population of free SpoVM, that first protein, goes rapidly downwards. It rapidly decreases. But you get the concomitant increase in the amount of bound SpoVM, but then very rapidly that population also decreases as the third population takes over. So this is going to be SpoVM and SpoVM that are bound to the surface of the membrane. And this population dominates in the first fraction of a millisecond. If we run the simulation for longer, what we notice is that this situation is quite stable for the first 15 minutes or so. But as you notice, at the end of the first 15 minutes, this population starts to go downwards, and a fourth population comes up, and this is going to be polymerized SpoVM that's on top of the SpoVM. And what we notice is that within a half an hour, this population dominates, where you get the SpoVM polymerized on top of the pre-localized SpoVM, and that stably holds everything into place. And so translated to in vivo, this is what we think is happening, where the proteins are both made in the mother cell cytosol. Once they get localized onto the forespore surface, polymerization begins, and eventually everything is locked into place by the time you get spore maturation. Okay, so you can tell me how I'm doing on time. If you want, I can definitely stop the talk there, or I can just give you the punchline of what we have for the drug delivery part of this.
You're at 27 minutes past the hour, so we kind of like it to,
We can wrap it up.
It depends if you have much to say about it. You can give us the punchline, and then we can get to some questions that people had in the chat and others that might
Yeah, how about I just give you the punchline? So I told you
Yeah.
that we can go ahead and assemble these particles using these two proteins on these membrane-coated beads. Someone told me that to publish this in a fancy journal, I had to give it a funny acronym. So we're going to call these shells. It's a tortured acronym that I won't tell you what it stands for. But the idea is that we could go ahead and modify the surface of these particles using a click chemistry technique. So I'm not going to go into this very much, but basically you can take your molecule of interest and click it onto the surface of these particles. And so what we decided to do was target a particular cancer type called HER2 positive breast and ovarian cancer cells that overexpress a particular receptor called the HER2 receptor. And what we decided to do was use a targeting moiety. It's a small peptide that binds to that receptor with a very high affinity. And so we took this little peptide and modified the surface of these particles with it. So this is the peptide that's been labeled here in red, and you can see that we can decorate the surface of these. And what we decided to do was to take those particles and then instead of using a solid silica bead, we decided to use a porous silica bead into which we can soak in a cancer drug called doxorubicin that fortuitously fluoresces. So this is a fluorescence microscope image of these doxorubicin soaked beads. We can then cover it with a membrane that's invisible in this image. We can then go ahead and then build that spore coat basement layer on top of it, and that's what's shown in green. And then we can go ahead and modify the surface of these with that targeting receptor. So I'll just leave it at this. I'm not going to give you the actual mechanism, but when you now administer in a mouse model for tumorigenesis, so this is actually, I'll go here, and tell you that we use this ovarian cancer cell line where you can introduce this tumor into mice. When the tumor reaches a certain volume, we go ahead and inject the mice with the free drug or with the drug encapsulated in our artificial spores, and then we measured the tumor volume over time. So when we did this with a lower amount of the doxorubicin drug, what we noticed is that the untreated tumors got bigger over time. If you treat the tumors with the free drug shown in red, the tumors reduced and didn't grow as fast, so that was great. But if you come back, what's shown here in green is the doxorubicin encased particles at the same concentrations. They do a much better job at controlling the tumor size. We did this at a second trial where we even increased the amount of doxorubicin, where you can see that the tumor size does a better job with the free drug, but still, even at this higher concentration, the particle encased doxorubicin does a much better job at containing the tumor volume. And what's not shown here is that the side effects that you get by injecting this concentration of doxorubicin at the site of injection is reduced when we use the particle enclosed doxorubicin. And these are just right before lunch to show you what these tumors look like. The point is that the tumors don't get better. So I'm not going to get into the actual data for this, but I will show you that this is the model where the particles we think bind to the surface. They're trafficked to an intracellular vacuole, where we've shown that the doxorubicin is released when the pH drops over here, and then it soaks out into the cytosol, where it's able to affect its activity. This is just a different model for brain metastasis in a zebrafish that we also showed where it was efficacious. And so we think that this might be an interesting additional strategy for the targeted delivery of drugs to specific sites. So I'll just conclude with 2 slides telling you that from the basic science part of today's talk, the basement layer of the spore coat contains these 2 unusual proteins, SpoVM and SpoVM. One recognizes membrane curvature at a micron scale. The other one is an oddball cytoskeletal protein that requires ATP hydrolysis to polymerize irreversibly. And we can reconstitute this whole thing in vitro to form these synthetic spore-like particles called shells. And then on the applied science part of this, we've shown that we can covalently display multiple proteins and small molecules of interest. We had one report where we suggested we can use this as a vaccine delivery platform. I didn't show you that data today. Some of the newer data suggest that we can actually use this as a tissue-specific drug delivery vehicle to specific cancer cells. So I'll end it there. So I told you all the people that have done the work. These are the people that have come through the lab so far. And all of the collaborators were acknowledged on the slides. And thank you for listening.
Thank you so much, Kumaran. That was a really fascinating talk. I loved it, where the basic science meets the applied. Awesome job. So we are at time, so those that have to leave, please do so. Don't feel bad. But those that can stay, please stay. And there are, if you have a few moments, Kumaran, there's a few questions. And I'd like to open the floor up to trainees first. So I was looking back in the chat. Arifa had a question from earlier, but she has a more general question. So go ahead, Arifa. Go ahead and ask. Are you still there, I hope? Yeah.
Hi, Dr. Ramamurthi. Thank you for that excellent talk. So I was just wondering about a very basic question. So you mentioned earlier on that the coat is actually very proteinaceous, the spore coat. So how exactly do these proteins, how are they able to withstand, extreme temperatures or conditions?
Right. So there are actually two layers to the spore surface. The second layer I didn't talk about, but it's also an active area of research in the lab. So there's the protein outer surface, and this is for Bacillus subtilis. This is going to be the outermost layer, the cell surface of the spore. There's an inner layer called the cortex. So that is a peptidoglycan layer. It's almost equally as thick as the coat. It's very thick. So these two layers combined are the ones that give you different resistances to different kinds of insults. So the coat is supposed to primarily give you resistance to things like chemical assaults and enzymatic assaults, things like lysozyme. And then the cortex is involved in keeping the dehydrated nature of the spore core. So the inside of the spore is actually crystalline. There's hardly any water involved at all, and the cortex is involved in keeping that almost water-free. So it's that cortex is actually directly responsible for high heat resistance. There's an interesting interplay here in that that cortex on the inside will not form unless you properly form the outer coat. And so how those two spatially unlinked structures are linked during development is actually an active part of research in the lab now too.
All right, thanks so much.
Sure.
All right, there was a question from Darren. Darren, did you want to ask a question or did you get it answered earlier?
Yeah, sure. Thank you so much. That was a great presentation, Professor.
Thank you.
I apologize, I joined a little bit late, but I was curious about the synthetic membrane model that you were using. Is it similar to the lipid nanodiscs developed by Dr. Stephen Sliger?
Yeah, so no. So that is as far as I understand, is not a supported system. So you actually have the protein on the outside and it's sort of a rubber band that's holding the lipids, right? So what we've done here, this is a supported lipid bilayer system. So many people are familiar with planar lipid supported bilayers. So there you have like basically a microscope slide that's perfectly flat, and then you put a double membrane layer on top of it. This is basically the same thing, except it's wrapped around a bead. Functionally, you build them almost the same way. So instead of using a cover slip, you'll just use a silica bead. And then you can pretty much put whatever lipids you want on the surface. So what's usually used is DOPC, phosphatidylcholine. So you can build it that way on the surface. In our, most of our experiments, what we're doing is a synthetic E. coli mimic, where you have phosphatidylethynolamine, phosphatidylglycerol, and cardiolipin at a particular ratio.
Got it. Thank you so much.
Sure.
Are there any other questions from trainees? Please raise your hand. I'm not seeing any of those. Maybe one last question. I think Anne had a question earlier. Do you want to ask that still, Anne? About the shell alone.
Yeah, in your I guess I should put my video on. Yeah, thank you. That was a great talk. So the shells alone, it looked like they had an effect on the tumors. So do you have an idea?
Yes, that was, even though the slide went by very quickly, that was a very acute observation. Okay, I can hand wave. Okay, so here's what I think, right? So this was a athymic mouse but it can do a primary immune response. Okay, so those particles that we had were so it didn't have the drug, but it did have the targeting moiety. It is attractive to think that if you can get that particle to where the tumor is, that there might be a secondary effect that's not directly related to the drug delivery, but that could be toxic to the target. And that's what I think is going on.
Yeah.
And it'll be interesting to see, I don't know, if macrophages are recruited to that site, for example. Yeah.
Yeah, thank you.
Yeah. I do want to stress, though, that we don't think that it's a general toxicity. So usually what in the nanoparticle field, the issue is that sometimes the empty particle alone will have a tumorigenic effect, and that's actually a general toxicity of the particle. We don't think that that's what's happening over here because as far as we can tell, the particles themselves, they accumulate eventually in the liver, they're cleared, but we don't think that there's a general toxic effect because, for example, the mice don't lose weight in the absence of the drug.
Mark did have a question. Mark, do you want to ask?
Yeah, sure. On the last piece, it occurred to me that what was sort of left out in the story is whether or not SpoVM has its own differential affinity for curved surfaces versus flat surfaces.
Right. I can tell you this. So we have not actively pursued this, but I can tell you anecdotally that if you produce both proteins in a regular cell that's not sporulating, so there you only have a concave curvature of membranes, you can produce both of them, but the second protein will not be recruited to the concave surface. So that is cute. So we haven't started yet. So it would be interesting to see if this polymer does have an intrinsic affinity for coating convex surfaces. It's at the back of our minds, but we haven't pursued it.
Yeah, because I was struck that the difference between the 2 micron and the 8 micron bead, the K half, was not so great.
Yeah.
But these spores are smaller, of course. They're probably about half micron, and so there might be a big difference there. But then, in some sense, if your final locking layer itself has some affinity, then you don't need to have such a strong affinity at the closer to the membrane side.
Yeah, absolutely. So, so we haven't invoked that higher affinity. So initially we just did the measurement of the free protein interaction between each other. But yeah, that would increase that further. Number one. Number two, the copy number of these proteins is ridiculous. I mean, it's in the, I think, 25,000 or something per cell, or 50,000 per cell. So even though you have slight differences in the affinity, when you run these simulations en masse, it makes a very big difference because you have a ton of this protein. So yeah, I think that helps push it, number two. And number three, you have that mysterious cooperativity that also helps push this towards the forespore.
Great, beautiful talk, thank you.
Thanks.
Last and final question, if you're up for it. Tom had one
I'm
question.
up for it.
Go for it, Tom.
Yeah, hey, thanks very much.
Sure.
That was really interesting. So I always like to think about how multicellular organisms stole things from microbes. And so I'm just curious if there are any close cousins to the SPO proteins in eukaryotes.
Yeah.
Sorry if you already went over that. I had to go.
I did not, but I'm smiling because we have a paper in revision right now that talks about the evolutionary history of SpoVM. This is that second protein. Okay, so it's an ATPase, right? It's a bona fide ATPase. If you look at the phylogeny of this protein, though, it totally belongs to a larger class of GTPases called the traffic GTPases. Okay, so if you look at all the P-loop GTPases in biology, there are two large classes. There's the traffic class and the CMIB class. So in the traffic class, you have proteins like RAS, dynamin, septins, ribosome-associated proteins that are in bacteria called ERA. SpoVM is evolutionarily derived from this ERA subfamily of proteins. So this is a ribosome maturation factor. It's a GTPase. What we think happened was that there was a gene duplication event at some point where you get two eras. SpoVM then picked up two additional domains and then went from being a GTPase to being an ATPase for evidently no good reason. Okay, so we wanted to know, just for kicks, what would happen if you resurrected its ancient GTPase activity, and why is that so, such a problem? So we did that, and that experiment actually worked in vitro. You can get the protein to more specifically hydrolyze GTP versus ATP. So that was amazing. So now at that point, we could do the experiment where we thought, okay, energy is energy. If you hydrolyze ATP, it doesn't matter GTP, you're going to release 30 kilojoules per mole. It should still work. It didn't. Okay, so in vivo, where you have ATP and GTP around, and this protein was able to promiscuously bind both, the protein was functional for sporulation, but in vitro, it wouldn't polymerize if you gave it just GTP. Okay, right. So the long story short is that we think that that's where that ADP-bound intermediate gets created, and the protein has evolved to specifically bind to ADP rather than GDP, and you need that to polymerize. Okay, and we think that the evolutionary reason for this was because there's some evidence in the literature that the amount of ATP stays relatively constant during sporulation, but GTP levels actually go down. And so we think that this protein switched its specificity. The reason I'm saying all this is because there's a very closely related protein to this family called septins that also require a GDP intermediate when they assemble. So this is an understudied class of cytoskeletal proteins. And so that is where I think we can get some insight into how that protein works by studying SpoVM.
Cool.
Mm-hmm.
When you had it in the GTP form, any chance you threw some unpolymerized actin monomers in a tube to see if it could
cheat and get in, or
see if it would interact with actin?
We haven't done that experiment.
I think with that, I think we'll give Kumaran a break. Thank you so much. It was an awesome talk.
That was a lot of fun.
Thank you so much.
Thank you. See you later.
Take care. Bye now.
Yeah, thanks. That was really entertaining. Appreciate it.